All right, good morning everyone. Audible? Start with biochemistry. Excited? No, not at all. So, yes, biochemistry is a very volatile subject, right? No matter how good you have learned it, I know most of you have forgotten most of the things. So, we'll do a quick revision together. Let's get started. Carbohydrate chemistry and metabolism, followed by lipid and then proteins. In the end, we will be doing certain miscellaneous topics and molecular biology. So let's get started. So as far as carbohydrate chemistry is concerned, we divide the carbohydrate into monosaccharide, disaccharide, oligo and polysaccharide. I'll tell you the important questions here. You should know the heteropolysaccharides. So the heteropolysaccharides are also called as mucopolysaccharides and are also called as glycosaminoglycans. So this here, we do not have to remember all the types of mucopolysaccharidoses. Just remember that mucopolysaccharides is a repeating amino sugar and an acidic sugar. It is a repeating amino sugar and an acidic sugar. Then isomers, there are various types of isomers. I'll focus on the important one. You have here anomers. The anomers are, if you see the structure of glucose, the structure of glucose has hydroxyl group at carbon atom 1. If the position of that hydroxyl group is below the plane of ring, it is alpha, and if the position of the hydroxyl group is above the plane of ring, it is beta. And the importance here is in digestion. We can digest the alpha linkage, we cannot digest the beta linkage. So we only digest alpha linkages. So the cellulose is having beta linkages, and because it is having beta linkages, this here cannot be digested, and hence it will form an important part of dietary fibers. Important part of dietary fibers. Then the next type of isomerism, one was alpha and beta anomerism. Another type of isomerism that exists is epimerism. In epimerism, you should remember the carbon atom which will decide the epimer. For example, the carbon atom 2, if it is different from that from glucose, it is called mannose. The only difference between glucose and mannose structurally, nobody will ask you the structure, do not worry. Just remember the carbon atom. Glucose and mannose, the only structural difference is carbon atom 2. And if here at carbon atom 2 the position of hydroxyl group in glucose is right, and in case of mannose it is left. So that is the only difference. Similarly, galactose. Galactose is also an epimer of glucose. You will have to compare each structure with glucose. So at carbon atom 2, if there is a difference, then we call it mannose, and if the difference is only at carbon atom 4, then it is called galactose. Which of these sugars is used as liver function test? Of all the sugars you know, which is used as liver function test? Galactose. It is this galactose which is used as liver function test. Why? Why do you use galactose? Because it is metabolized only in liver. So if something goes wrong with the liver, its concentration will increase, right? Then the next type of isomerism is enantiomers. The next type of isomerism is enantiomer. Here also the change is only in the position of hydroxyl group. but who decides? At which carbon atom it is deciding? For example, अभी हमने थोड़ी देर पहले अभी epimerism देखा था, उस पे carbon atom 2 and carbon atom 4 was epimer of glucose and mannose and galactose was formed, right? Here the deciding factor is the penultimate carbon atom. If the deciding factor is the penultimate means the second last carbon atom for glucose. First C for glucose. In glucose, at the penultimate, that means the second last carbon atom, 1, 2, 3, 4, 5, 6, note the change at the fifth carbon atom. If the position of hydroxyl group is on the right hand side, then we will call it capital D glucose. If everything is same, the position of hydroxyl group is on the left hand side, then we will call it capital L glucose. So here this one is D and this one is L, the positioning of hydroxyl group according to that. So if the position is on the right hand side for the hydroxyl group, then it is called capital D, and if the hydroxyl group is on the left hand side, then we call it capital L. Do not confuse capital D and capital L with small d and small l. Small d is for dextrorotatory and small l is for levo-rotation. वो totally different है. That is on the basis of optical rotation. This is simply on the basis of structure. The first time when the enantiomerism was studied, it was studied for glyceraldehyde, and that is what you can see here above. We have given a comparison of glyceraldehyde, and that is also one of the questions, that which is the parent molecule, because the most common wrong answer for this is glucose only, right? So the first time it was studied for glyceraldehyde, so glyceraldehyde is called parent molecule for, it is considered parent molecule for isomerism, for the study of isomerism. So that was the difference between enantiomerism, epimerism and anomerism. So the deciding factor for anomerism is which carbon atom? 1 for glucose and 2 for fructose. Note this down. The deciding factor for anomerism is carbon atom 1 for glucose and carbon atom 2 for fructose. If the alpha is below the plane of ring, we will call it alpha glucose. If the hydroxyl group is above the plane of ring, we will call it beta. Then who decides the epimerism? It is individually discussed at each carbon atom. For this you just have to remember 2. Carbon 2 change in glucose will result in the formation of mannose, and carbon 4 change in glucose will result in the formation of galactose. And for enantiomerism, the deciding carbon atom is penultimate carbon atom. For glucose it is 5th carbon atom, and for glyceraldehyde it will be which carbon atom? Carbon atom 2, because glyceraldehyde is only 3 carbon atoms. So in it the penultimate carbon atom is 2nd carbon, right? Then this is, now we are starting with metabolism. Those were the few important questions in the carbohydrate chemistry. Besides this, what is fruit sugar? Fructose. Fructose is coming from which type of sugar? It is coming, what is fructose coming from? It is coming from sucrose. And what is sucrose? It is glucose and fructose. It is glucose and fructose. What is the source of sucrose? It will be, it is the normal white sugar which we eat, the cane sugar. That is the sucrose. So sucrose will, on the hydrolysis, it will split to form glucose and fructose. Then what is milk sugar? The lactose. And lactose splits to form glucose and galactose. So those were minor questions. Not many questions are there from carbohydrate chemistry. So I've just gone through it. The next topic is important, and that is the metabolism of glucose. We'll start with glucose transporters. There are 5 major transporters you should know: GLUT1, 2, 3, 4 and 5. Please note, first GLUT1, see where it is present. It is present in brain, kidney, colon, placenta, and this is RBC, right? So this is red blood cells. So the function of them is glucose uptake. The function of here is glucose uptake. Similarly, GLUT1 के साथ study GLUT3. The GLUT3, if you see, it is mainly similar type of tissue distribution जैसा GLUT1 का है. The only difference here is of RBC. The RBCs mainly have or only have GLUT1. They do not have GLUT3. Then GLUT5. GLUT5 is present in small intestine and is required for the fructose metabolism. It is uptake of fructose which is helping, it adds fructose. It is in small intestine and sperms. These are the two tissues which are having GLUT5. So GLUT5 is not a glucose transporter but is rather a fructose transporter. वो एक misnomer है, and that is why it is most commonly asked. Then out of all these transporters, GLUT2 and GLUT4 is most important. So let's here see GLUT2 and GLUT4. Always asked in comparison. Which is the bilobed organ in our body? I suppose my anatomy is not that very good, but I will still say liver. अगर आप उसका third and fourth smaller lobules छोड़ दो, by and large liver is a bilobed organ. I use it to remember that liver has GLUT2. So liver is having GLUT2. Besides liver, kidney, we have 2 kidneys. So kidney is having GLUT2. Small intestine has GLUT2. Out of 2 and 4, the smaller number is 2. So small intestine has GLUT2 and pancreas has, yes, pancreatic beta cells is having GLUT2. Which is the 4-chambered organ in our body? Heart. We all agree unanimously. God specifically made it 4-chambered so that we can remember it has GLUT4. So this is heart. Besides heart, GLUT4 is present on, yes, skeletal muscle and adipose tissue. Adipose tissue. These are the major sites. I write 2 like this. It helps me remember that GLUT2 is insulin independent. That makes GLUT4 insulin dependent. क्योंकि comparison में याद रखना है. So you have to remember that GLUT2 is insulin independent and GLUT4 is insulin dependent. Then the Km. The Km for GLUT2 is 15 to 20 millimoles and the Km for GLUT4 is just 5 millimoles. Which has higher Km? Clearly it is GLUT2. Then that means that GLUT2 requires more glucose to reach its maximum velocity. What is the affinity for glucose for GLUT2? Affinity क्या है फिर? As compared to GLUT2 and GLUT4, which is having more affinity for glucose? GLUT4 is having more affinity. Why? Because उसका Km कम है. That means it needs only small concentration to reach Vmax. Right? So अगर उसको थोड़ा सा concentration चाहिए velocity maximum reach करने के लिए, इसका मतलब उसका affinity ज़्यादा है. On the other hand, GLUT2 requires a lot of glucose to reach its velocity maximum. That means it does not like glucose that much. Right? So that was GLUT2 and GLUT4. GLUT2 and GLUT4. Now, some important questions from glycolysis. So far what we have seen is that glucose has entered inside the cell. भले वो pancreas हो, liver हो, भले वो कोई भी tissue हो, brain, kidney, whatever. So once the glucose enters in the cell, in the extrahepatic tissues, the majority of glucose, the only function is that it will enter into glycolysis. Majority of glucose will be entering into glycolysis. So in glycolysis, this is complete glycolysis, converting glucose to pyruvate. Glucose is the converted to pyruvate, but you do not have to remember all these steps. You just have to remember 3 irreversible steps. So you tell me what are the 3 irreversible steps? Number 1, the first step, glucokinase and the hexokinase step. Mark those. I am just telling you what you have to remember. So the first step, glucokinase, hexokinase, is the irreversible step. The second is PFK1 step, phosphofructokinase step. That is the second most important or the rate limiting enzyme. And the third is pyruvate kinase step. Out of all the 3 steps, the most important step for glycolysis is PFK1, phosphofructokinase 1. So these are the 3 irreversible steps of glycolysis. Then the difference between glucokinase and hexokinase. Please note the difference between glucokinase and hexokinase. So here by and large we say that glucokinase and hexokinase is same, but no. It has different tissue distribution. Like GLUT2 and GLUT4 were different, similarly glucokinase and hexokinase are different. The major difference is glucokinase is present only in liver. Hexokinase is present in extrahepatic tissues. The glucokinase has a Km of 5 millimoles. Glucokinase has a Km of 5 millimoles, but hexokinase has a Km only of 1 millimole. So which has more affinity for glucose? Clearly hexokinase. Clearly hexokinase. Then these are some additional points, that glucose 6-phosphate inhibits hexokinase. Glucokinase is not inhibited by glucose 6-phosphate. So that was 3 important steps of glycolysis, 3 irreversible steps. इसके अलावा और सारे steps are reversible. I am not even discussing them, right? But these are the 3 most important steps. And then inhibitors of glycolysis. The fluoride inhibits the enzyme enolase, and this is the most important inhibitor. Whenever you take blood glucose for estimation, you take it in a vial which is having grey colored top. Worldwide you take it in a grey colored top. That signifies that this vacutainer is having sodium fluoride and potassium oxalate. The vacutainer is having sodium fluoride and potassium oxalate. Then the next point related to glycolysis is there are 3 substrate level phosphorylation total in biochemistry. The substrate level phosphorylation means these are those steps where ATP is formed. ADP gets converted to ATP. These 3 steps which are substrate level phosphorylation are: number 1, phosphoglycerate kinase, number 2 pyruvate kinase, and number 3 is not in glycolysis but is an enzyme of TCA and is succinyl CoA thiokinase. Succinyl CoA thiokinase. So these are the 3 substrate level phosphorylation. Please note these are not major source of energy. The major source of energy comes from oxidative phosphorylation. उसमें पहले इलेक्ट्रॉन ट्रांसपोर्ट चेन होगा और फिर ऑक्सीडेटिव फॉस्फोराइलेशन होगा. द वे वी से NADH forms 2.5 ATP, FADH2 forms 1.5 ATP. That is the major source of energy. This is minor source of energy. ठीक है. So these 3 substrate level phosphorylation, the 2 enzymes are present in glycolysis. One enzyme we will see in TCA cycle. And when we will see in TCA cycle, I will ask what was the first 2 enzymes. तो इनको याद करो. Phosphoglycerate kinase and pyruvate kinase. Those 2 kinases are substrate level phosphorylation in glycolysis. Then coming to the clinical correlation. Please note that hemolysis, I suppose you all remember that number 1 cause of hemolytic anemia is G6PD deficiency. Worldwide it is the most common enzyme deficiency and it is the number 1 cause of hemolytic anemia. However, the number 2 cause of hemolytic anemia under enzyme defects is pyruvate kinase deficiency. So nowadays they are not focusing that much on G6PD deficiency because mostly students remember that it is the number 1 cause of hemolytic anemia, and that is why they have started asking questions on pyruvate kinase deficiency, right? Also, I will tell you each pathway ka most important rate limiting enzyme. You should know everything about that rate limiting enzyme, right? No, I am not saying that direct question will be asked ki which of the following is the rate limiting enzyme, but still the question is framed in such a way ki answer is still the rate limiting enzyme, right? So every pathway you should know. If I tell you that this is the rate limiting enzyme, you should know everything about that enzyme. For example, in glycolysis, the rate limiting enzyme is, what is the rate limiting enzyme for glycolysis? PFK1. That is the most important rate limiting enzyme, right? So now Rappaport Lubering shunt, it forms 2,3-DPG. And what is the significance of 2,3-DPG? It shifts the hemoglobin saturation curve to the right. More important from physiology point of view, but yes, one thing which you must remember is that where it is coming from. It is coming from glycolysis in RBC, only in RBC. So this Rappaport Lubering shunt occurs in RBC and it forms 2,3-bisphosphoglycerate. The function of this is to shift the hemoglobin saturation curve to the right. And this is what happens in the presence of 2,3-DPG. That means more oxygen delivery to the tissues. More oxygen delivery to the tissues. So that was glycolysis. Rate limiting enzyme for glycolysis? PFK1. You must remember the 3 irreversible steps of glycolysis and the 3 substrate level phosphorylation, and the most important is the inhibitor of glycolysis, and that is enolase is inhibited by sodium fluoride. Sodium fluoride. The next is the link reaction, and this here is PDH. Here is a very important question on PDH. PDH requires 5 coenzymes. Can you see it on page number 7, the first thing? What are the 5 coenzymes? PDH requires pyruvate dehydrogenase is the link reaction between glycolysis and TCA. This PDH requires thymine, lipoic acid, coenzyme A, FAD and NAD. I use the mnemonic tender love care for Nancy. That is how I remember those 5 coenzymes required for PDH. Thymine, lipoic acid, coenzyme A, FAD and NAD. What is thymine? Which vitamin? B1. What is lipoic acid? It is not a vitamin. That was just to wake you up. Then what is FAD? Vitamin B2. Riboflavin forms flavin adenosine dinucleotide. What is vitamin this niacin coming from? Niacin forms, the active form of niacin is NAD, nicotinamide adenine dinucleotide. Then the coenzyme A comes from which vitamin? B5. It is the active form of vitamin B5. In alcoholics, now this is important. Alcoholics are very important. In any exam, alcoholics are most important. Right? So the topic, not alcoholics as such. So this vitamin, in alcoholics, few vitamin deficiencies are more common than others, like B1, B6, B9 and B12. These are few vitamin deficiencies which are more common. होने को कोई भी vitamin deficiency हो सकती है, but these are the most common vitamin deficiencies found in alcoholics. Out of which B1 deficiency is most common, and B1 deficiency in alcoholics results in Wernicke-Korsakoff psychosis. Yes, Wernicke-Korsakoff psychosis. The Wernicke-Korsakoff psychosis is characterized by Goa, not the place from where they became alcoholic. Here it is mnemonic to remember global confusion. They are so confused that they start forming their own stories. Mangadat kahaniya, that is coming as a result of confabulation. O, ophthalmoplegia and A, ataxia. So under PDH, you should remember that PDH requires 5 coenzymes, out of which thiamine is most important, and because thiamine is required by PDH, vitamin B1 deficiency will result in low in energy, because glucose ka major role in our body is to produce energy. So it will be resulting in low energy. So this was due to vitamin B1 deficiency in alcoholics. We will be discussing this again under vitamins also. Then anaerobic glycolysis. What is the end product of anaerobic glycolysis? See here, it's there on your notes. It will form lactate. The tissues which form the lactate are RBC. There are 2 types of muscle, red and white. It is white muscle fibers which perform anaerobic glycolysis. Lens, retina, brain, renal medulla. But the first 2 most important. The next is the link reaction goes further, enters into TCA. So far we are discussing aerobic glycolysis. So aerobic glycolysis, first step is conversion of glucose to pyruvate. Then pyruvate gets converted to acetyl-CoA by the enzyme PDH, and PDH was requiring 5 coenzymes. PDH is a step in the mitochondria, and so is TCA. So TCA also occurs in the mitochondria, and hence it does not occur in, obviously it does not occur in cytosol agar mitochondria mein ho raha hai, but which tissue does not have mitochondria? RBCs. So it does not perform, and hence the only source of energy for the RBC is anaerobic glycolysis. They can never perform aerobic glycolysis. The tissue whose major source of energy is only aerobic glycolysis is? You don't love your brain. Brain eats only glucose and performs only aerobic glycolysis. Unless and until somebody goes for prolonged starvation. After 2 to 3 days of starvation, now brain will start using? Yes, ketones. It will start using ketones. So, the TCA is a amphibolic pathway. It's written here. It occurs only in mitochondria and gives 10 ATP per cycle. It gives 10 ATP per cycle. This is how they calculate the 10 ATP. You should remember all the steps of TCA. Glycolysis mein I told you, do not remember all the steps. Just remember the 3 irreversible enzymes. But here for TCA you should try and remember all the steps, because it's very easy to form an image based question. What they will do is, they will form the circle, put some intermediates, remove some other intermediates, and then they can ask which is the product. So here this is quite important. I know it is overwhelming, but it is quite important. So what will glucose form? Ultimately glucose forms pyruvate, and pyruvate forms acetyl-CoA. Try to locate acetyl-CoA here. Acetyl-CoA is a 2-carbon molecule which combines with 4-carbon oxaloacetate to form a 6-carbon citrate. 6-carbon citrate. Citrate will then form isocitrate. Isocitrate will then form alpha ketoglutarate. I am pointing the important ones which you must remember. Alpha ketoglutarate forms succinyl-CoA. Succinyl-CoA succinate, fumarate, malate, and then finally oxaloacetate. So now here one important thing. Remember we said there are 3 substrate level phosphorylations. The first 2 substrate level phosphorylation we have already seen under glycolysis. This here, note at succinyl-CoA thiokinase step. It is converting ADP to ATP, and hence this is the third and the last substrate level phosphorylation. The total energy produced by anaerobic glycolysis is, note somewhere here only, anaerobic glycolysis will form only 2 ATP. The aerobic glycolysis will form 32 ATP. So it is a huge difference, and the TCA cycle, per cycle it forms only 10 ATP. So that is the energy released from glucose under aerobic and anaerobic condition, and the energy released from the TCA. This is gluconeogenesis. I will tell you the important things to remember. This is the most important slide under gluconeogenesis. There are 4 substrates for gluconeogenesis. See, once we have covered glycolysis now. Glycolysis, here we have the process of converting glucose to carbon dioxide and water and generates lot of energy. That is aerobic glycolysis, complete aerobic glycolysis. Now that glycolysis is reversed in gluconeogenesis, but it is only reversal from the formation of glucose to pyruvate. So pyruvate will be converted back to glucose. For this you will require gluconeogenic substrates. The gluconeogenic substrates are 4. Number 1, amino acid. Number 2, lactate. Number 3, propionate. And number 4, glycerol. So I am marking the most important topics under gluconeogenesis. You have to remember the 4 substrates, and out of this 4 substrate, the most important substrate is glucogenic amino acids. Glucogenic amino acids. The key enzyme of gluconeogenesis. What were the 3 enzymes of glycolysis? By now अब याद हो जाना चाहिए. First glucokinase, hexokinase. To reverse that, here you can see the 4th enzyme, glucose 6 phosphatase. That is reversal of glucokinase. What was the second enzyme? PFK1. PFK1 is reversed by the 3rd enzyme written here, that is fructose 1,6 bisphosphatase or diphosphatase, one and the same thing. And the last step was pyruvate kinase. And to reverse pyruvate kinase step, you require 2 enzymes. One is pyruvate carboxylase and the second one is PepsiK, phosphoenol pyruvate carboxykinase. So these are the key enzymes of gluconeogenesis. If I have to pick one from all of them, I will say that fructose 1,6 bisphosphatase is the most important step. So this here is the most important step, fructose 1,6 bisphosphatase. So that is the rate limiting enzyme. Note the rate limiting enzyme for each pathway. Then glycogenesis and glycogenolysis. The rate limiting enzymes only. What is the rate limiting enzyme for glycogenesis? It is glycogen synthase. And what is the rate limiting enzyme for glycogen breakdown? It is glycogen phosphorylase. Please note, the glycogen phosphorylase requires vitamin B6. Vitamins are very important topics. We'll be discussing them in a while. Very, very, very important. So this glycogen phosphorylase requires vitamin B6. The vitamin B6 is therefore required for glycogenesis or glycogenolysis. Genolysis. So this here is the rate limiting enzyme for glycogenolysis. Here in this pathway, another important thing is that UDP glucose is the starting substrate to form glycogen. So UDP glucose is the starting substrate. It is starting substrate to form glycogen. If they ask you which is the starting substrate to form glycogen or which of the following forms of glucose will ultimately form glycogen, it is UDP glucose. And in which form is glycogen broken down to? The answer is glucose 1-phosphate. Please note here, the ultimately glycogen is broken down to glucose 1-phosphate. So this here glucose 1-phosphate is the end product of glycogen breakdown. It is the end product of glycogen breakdown, because there are questions like which of the following forms of glucose is released from the breakdown of glycogen. So the answer is glucose 1-phosphate. The last reaction here is glucose 6-phosphatase. Glucose 6-phosphatase will convert glucose 6-phosphate to free glucose. It will convert glucose 6-phosphate to free glucose. This enzyme glucose 6-phosphatase is present only in liver. This enzyme glucose 6-phosphatase is absent in muscle, and hence although we have 2 types of glycogen, liver glycogen and muscle glycogen, only liver glycogen maintains blood glucose. जो भी glycogen is getting stored in the muscle, that will be used by muscle for its own activity. It will be not raising the blood glucose. Why? The answer is glucose 6-phosphatase is absent in muscle. Muscle do not have this enzyme glucose 6-phosphatase, right? Because liver is the only tissue which is having this enzyme, hence liver is the only one which maintains the blood glucose. And this is the most important topic under carbohydrate metabolism, and that is your glycogen storage disease. Put this under last minute revision topics. This should be put under last minute revision topics. You will always find a question on glycogen storage disease. Type 1 is also known as von Gerk's disease. Von Gerk's disease is deficiency of the enzyme glucose 6-phosphatase. Please do not confuse glucose 6-phosphatase with G6PD. G6PD is an enzyme of HMP shunt. ये glucose 6-phosphatase is an enzyme of glycogenesis. ठीक है? Glycogenolysis more appropriately. So this enzyme is important for glycogen metabolism. G6PD is an enzyme for hemolytic anemia, deficiency of whom is causing hemolytic anemia. बहुत क्योंकि एग्जाम में क्या करते हैं वो, they will not write the full form for glucose 6-phosphatase. They will write G6PASE. And most of the students, they will confuse between G6PASE and G6PDH or G6PD. So ये बहुत common confusion है. So this glucose 6-phosphatase deficiency results in von Gerk's disease. If somebody is having G6PD deficiency, it results in hemolytic anemia. So glucose 6-phosphatase deficiency is causing fasting hypoglycemia. 5 features you must remember. Number 1, fasting hypoglycemia. Number 2, lactic acidosis. Number 3, you can add here hyperuricemia. And number 4, ketosis. And number 5, hyperlipidemia. So those are the 5 cardinal features of von Gerk's disease. Do not confuse von Gerk's disease with Cory's disease. Cory's and von Gerk's, they both present with similar feature. Cory's is type 3 glycogen storage disease. अच्छा, Cory's and इसमें another differentiating factor is lactic acidosis, which is present here but absent in Cory's. And then hyperuricemia, which is present here but absent in Cory's disease. So Cory's will present similar to that of von Gerk's, except there is no lactic acidosis and no hyperuricemia in Cory's. So type 2 Pompey's disease. Pompey's is the only glycogen storage disease which affects lysosome. So this enzyme here is lysosomal acid maltase. If this enzyme is defective, it will result in 2 major symptoms. One is muscle weakness and another is heart failure. So in Pompey's, the cause of death is heart failure. Remember pump failure. That way you will remember. It is a very poor prognosis disease. It causes death by 2 years of age. Very poor prognosis disease. Then we have type 3. Type 3 is Cory's. I have already discussed Cory's का same presenting feature as that of von Gerk's disease. But the enzyme is different. For Cory's, the enzyme is defective D-branching enzyme. Defective D-branching enzyme. Write down mnemonic here to remember this. Cory's can't cleave. And because they cannot cleave, they do not have D branch. Anderson's can't add. Therefore, they do not have branching enzyme. So write down here, Anderson's can't add. That should help you remember that Anderson's do not have branching enzyme. So even this is very poor prognosis disease. Anderson's causes death by 5 years of age. Like Pompey's was poor prognosis, causing death by 2 years. Anderson's is also poor prognosis. McArdle's is only affecting muscle. So that is easy to remember, MEC-MS. So it is deficiency of muscle phosphorylase in McArdle's disease. And if somebody does not have muscle phosphorylase, of course these people, they don't like to move their muscles a lot. So it is defective. If they do not have muscle phosphorylase, it will result in exercise intolerance. इससे पहले देखो मोस्टली फीचर क्या है, फास्टिंग हाइपोग्लाइसीमिया, बिकॉज लिवर ग्लाइकोजन वॉज अफेक्टेड. हियर इट इज मसल विच इज अफेक्टेड. देन हर्स इज लिवर फॉस्फोराइलेस. राइट डाउन हियर लिव हर. That way you will remember hers is defective liver glycogen phosphorylase. The mnemonic is live her. So that way you should remember that liver is defective in Hers disease. So it is liver glycogen phosphorylase defect. And the last one, Thyroid disease, is same as that of McArdle's. Same presentation, PFK1 deficiency as that of McArdle's. There also we have written that it is type 5, but there is one additional finding here, and that is hemolysis. And that is hemolysis. So that was glycogen storage disease. In HMP shunt, I mainly want you to remember, most important point here is two things. One is your HMP shunt is giving NADPH by the enzyme G6PD, and whose deficiency results in hemolytic anemia. By the way, NADPH is how much energy in our body? One NADPH gives how much ATP? Yes, 2.5 ATP is the most common wrong answer. It gives 0 ATP. It does not give any energy. It is NADH which gives 2.5 ATP, not NADPH. Another thing which I want to remember is, here you can look at transketolase. This enzyme, transketolase, requires vitamin B1. This enzyme is so important for vitamin B1, or rather vitamin B1 is so important for this enzyme that if there is deficiency of B1, it will decrease the activity of transketolase. And hence we use erythrocyte transketolase to know somebody's vitamin B1 status. Very important question. Which enzyme activity do we use to note B1 status of an individual? The answer is RBC transketolase. पूरे पाथवे में वो दो सबसे इम्पॉर्टेंट पॉइंट्स हैं. The next is NADPH significance. The NADPH is required for the formation of reduced glutathione. It forms reduced glutathione. The reduced glutathione requires two enzymes. One is glutathione reductase and another is glutathione peroxidase. Can you note the coenzyme required for glutathione reductase? What is the glutathione reductase requiring? FAD. And what is glutathione peroxidase requiring? Selenium. Right? Both of these are important questions. Glutathione reductase requires FAD and peroxidase requires selenium. Selenium and glutathione peroxidase both are antioxidants. Both of them are antioxidants because they are required by this enzyme here. Another thing here is, like we discussed for transketolase. Transketolase के लिए most important vitamin was B1. Similarly, for glutathione reductase, the most important vitamin is B2. And B2 की activity के लिए we use this activity of the enzyme. So if I have to know somebody's vitamin B2 status, I will go for the activity of the enzyme glutathione reductase. Please note, you will get similar questions. Question will be framed like, which of the following is used to assess vitamin B2 status? Option A, glutathione peroxidase. Option B, glutathione oxidase. Option C, glutathione dehydrogenase. Option D, glutathione reductase. So what is your answer? Glutathione reductase requires. But can you note कि कितना similarly framed question था, right? तो अगर इतना भी याद रह जाएगा कि there was something gluta, अगर वो नहीं याद रहा कि it is glutathione reductase, still we are not able to solve the question, right? So what I mean to say is, in biochemistry they will always give similarly framed questions. Options हमेशा confusing होंगे. They'll try to confuse you between the options. पहले ही biochemistry volatile है, पहले ही वो बहुत दुखी करती है, और ऊपर से जो options है वो और दुखी करते हैं, right? So that is why this session is important, so that you know what are the important points here. So this glutathione peroxidase requires selenium and glutathione reductase requires vitamin B2, right? Then I'll just discuss the two important disorders related to fructose metabolism. Fructokinase, if deficient, results in essential fructose urea. That means this person is simply excreting fructose in urine. And essential fructose urea, no treatment is required. You do not treat this patient. You just reassure them that whenever you will get Benedict's test, get for your urine analysis, it will come out to be positive. But it is not going to, this is not going to go anywhere any further. So the metabolic pathway ends at fructose. So it was not a harm to you. So, but it will always give Benedict's test positive. If somebody does not have aldolase B, the disorder is called hereditary fructose intolerance. Hereditary fructose intolerance. In hereditary fructose intolerance, there is accumulation of fructose 1-phosphate. And here we will treat, because it is intolerance to fructose. This is usually a 6-month-old child who have suddenly developed vomiting, diarrhea, acute liver failure symptoms. Because at 6-month-old child, we start with fructose. We start weaning. And the first thing which we give is usually fruit juices. And that is caused intolerance. The child has immediately developed vomiting, diarrhea, acute liver failure type of symptoms, hyperbilirubinemia and all. So aldolase B deficiency results in accumulation of fructose 1-phosphate. Do we treat this? Yes, of course we treat it. This is not a harmless situation. This is harming child. So this treatment here will be no fructose. And what other sugar will you not give? Which other sugar was forming fructose? Sucrose. So we do not give this child sucrose also. So we do not treat essential fructoseuria, but we treat hereditary fructose intolerance. Similarly, the most important enzyme for galactosemia is, in galactosemia also, this enzyme. I just want you to remember the first and second enzyme here. Just one enzyme. And that is galactose 1-phosphate uridyl transferase. This is classical galactosemia. Deficiency of this causes classical galactosemia, and once this sets in, again the presenting feature will be same as that of hereditary fructose intolerance. वो सेम ही की तरह प्रेजेंट करेगा एज दैट ऑफ हेरिडिटरी फ्रक्टोज इंटॉलरेंस. Classical galactosemia presents same as that of hereditary fructose intolerance, except one major point. This will also have bilateral cataracts. They can also give you an image showing a child who is 1 month old and is having congenital cataracts, and that is your clue of galactosemia, classical galactosemia. We are on page number 15. Yes. The lipid metabolism. The next is lipid metabolism. Out of all the fatty acids, you must remember lauric and myristic acid. The lauric and myristic acid. This is present in coconut oil. The coconut oil is rich in saturated fatty acids. It is rich in saturated fatty acids, and that is lauric and myristic acid. Physiologically, most important oil is palmitic acid. The form of oil which we form in our body is palmitic acid. That is the hydrolyzed form. Carbon 16 palmitic acid. Then we have 3 essential fatty acids. These are linoleic acid, alpha linolenic acid, and arachidonic acid. Note the carbon chain. It is 18 carbon, linoleic, alpha linolenic is also 18, and arachidonic acid is 20 carbon. The number of double bonds in linoleic is 9, 12, 2. At alpha linolenic, the position of double bonds is 9, 12 and 15, 3 double bonds, and in arachidonic there are 4 double bonds at position 5, 8, alpha 4, 11 and 14. It's given in the slide only. 5, 8, alpha, 11 and 14. Now, which of these is omega 3 and omega 6 fatty acid? पता कैसे चलेगा कि कौन सा ओमेगा 3 है और कौन सा ओमेगा 6 है? All you need to do is, पहले देखो कितना कार्बन एटम्स है. For example, linoleic is how many carbon atoms? 18. What is the position of the last double bond? It is at 12. For linoleic, it is 12. So just minus 18 minus 12. So it will become omega 6. Similarly, for alpha linolenic, it is 18 carbon. The last double bond is present at carbon number 15. So it is 18 minus 15, 3. And similarly, we have arachidonic acid, 20 carbon atom. The last double bond is at 14. So it is omega 6. So omega 6, 3 and omega 6 fatty acid. Now, out of this, which is most important? The linoleic is most essential fatty acid. out of this, which is least important? Arachidonic acid is least essential fatty acid. And which is cardioprotective? Omega 3 fatty acid. Omega 3 capsules भी मिलते हैं. Nowadays we give supplements for omega 3 fatty acids because it is cardioprotective. And you have to remember the name of that, and that is alpha linolenic. They'll give you options like which of the following is cardioprotective fatty acid. So answer is alpha linolenic fatty acid. अच्छा, you have to remember this 5 names. Phospholipids के पांचों नाम you have to remember because the questions are like which of the following is a phospholipid except, all except. So all except में you will find options में lecithin भी हो सकता है, cephalin भी हो सकता है, serine भी होगा, phosphatidyl inositol भी होगा और cardiolipin भी होगा. इनमें से just remember that cardiolipin is present in the mitochondrial membrane of heart, and that is why the name cardiolipin, it's related to heart. Another thing is, and then there will be one other option which is not a phospholipid. कोई भी lipid डाल देंगे. For example, triacylglycerol is not a phospholipid. It's not phosphate containing lipid. So you have to remember names like that only. Now, what is the importance of lecithin? Clinically, why do we need lecithin? At 24 to 28 weeks of fetal life, the fetus start forming lecithin more than sphingomyelin, and there is a ratio known as L to S ratio. This ratio should be more than 2, and that is an assessment of the fetal maturity, that fetal lung is mature enough, because lecithin is responsible to decrease the surface tension. If lecithin is not there, surface tension बहुत ज़्यादा हो जाएगा and alveoli will collapse. And that is why if lecithin is absent, it results in IRDS, infant respiratory distress syndrome. And there is a question that what is lecithin? It is a type of phospholipid. So you have to remember the 5 names, and you must remember that lecithin, if deficient, results in infant respiratory distress syndrome or acute respiratory distress syndrome, and it is formed during the fetal life. So anytime preterm delivery has to be done, we should go for L to S ratio. Now, in addition to these phospholipids, I just want you to remember a 6th phospholipid, and that is a sphingolipid known as sphingomyelin. It is also a phosphate containing lipid, sphingomyelin. So options में जो भी इन 6 के अलावा दिया होगा, that will be your answer, because these are all phosphate containing lipids. And the next important topic is this. Put this under last minute revision topic. Like you have done for glycogen storage diseases, sphingolipidosis is very, very, very important. They are difficult to remember, that is why they are important. Anything which you will find difficult to remember, it will be there in the examination. It's not my wish, but it is how inevitably how it is. So this is sphingolipidosis. Let's discuss one by one, starting with Tay-Sachs. Tay-Sachs is deficiency of the enzyme hexosaminidase A. It is accumulating lipid is ganglioside. Just remember GM2. GM2 is the accumulating lipid in Tay-Sachs. In Tay-Sachs, you will get, it is a common topic in pathology also, where they'll talk about its biopsy. The biopsy gives onion skin lysosomes, right? And that is characteristic for Tay-Sachs. I haven't shown it here, but it is the pathological correlation. Tay-Sachs is exactly same in findings as that of Niemann-Pick. Can you note Niemann-Pick? I guess the second last disorder. In sphingomyelinase deficiency will result in Niemann-Pick disease. Niemann-Pick is similar to that of Tay-Sachs. दोनों में there is something known as cherry red spot on macula which is seen. दोनों में ही एक common finding है cherry red spot on macula. See, there are some findings like mental retardation, वो तो सभी में ही है, right? All of them are lysosomal storage diseases, वो सभी में ही है. But there are some characteristic findings which should help you make the diagnosis. Like cherry red spot in macula is seen in both Tay-Sachs as well as Niemann-Pick. But the differentiating feature is hepatosplenomegaly. In case of Niemann-Pick, there is hepatosplenomegaly. It is absent in Tay-Sachs. There is no hepatosplenomegaly in Tay-Sachs. The next important disease is Fabry's. Fabry's is defective alpha-galactosidase. It is the only X-linked disorder. All the others are autosomal recessive. Fabry's is only X-linked disorder. Then remember Gaucher's. Gaucher is deficiency of the enzyme beta glucocerebrosidase. And Gaucher is the only one where successful enzyme replacement therapy is available. So this here has enzyme replacement therapy. It's very expensive. I guess for 6 months it is somewhere 50 lakhs, but it is available. But the next one is recently they have found a drug for alpha-galactosidase, and that is Fabry's disease. That is Fabry's disease. Recently enzyme replacement therapy is possible for this also. But the first time ERT was done successfully was for Gaucher's. The next is Krabbe's. Krabbe's is deficiency of the enzyme beta galactosidase. Beta galactosidase. तो alpha galactosidase है तो enzyme defect, then the disease is Fabry's. But if this beta galactosidase, then the defect is Krabbe's. Gaucher's is which enzyme defect? Beta glucosidase or beta glucocerebrosidase. Both are same. Both are same. Right? It's here given sidase, but it is same as cerebrosidase or sidase. Now, sphingomyelinase defect is Niemann-Pick, and hexosaminidase A defect is Tay-Sachs. Tay-Sachs. See, no matter how well you understand this, you will forget it. So put this under last minute revision topic. Very important. It is important. Then just remember the rate limiting enzyme for lipogenesis, and that is your rate limiting enzyme for lipogenesis is acetyl-CoA carboxylase. Please note this enzyme is activated by citrate and is inhibited by palmitic acid. Also note that it requires biotin. Biotin is what? B7, also known as vitamin H. The next is NADPH is required for fatty acid synthesis. It is coming from HMP shunt. And how much energy is NADPH equivalent to? 0 ATP. Now you did not get trapped. Huh? The next is fat lipogenesis में वही एक important question है, that is acetyl-CoA carboxylase, right? The next is fatty acid oxidation. In fatty acid oxidation, I want you to remember activation में there is an enzyme known as CPT1. Activation of fatty acid requires an enzyme known as CPT1, and this is inhibited by malonyl coenzyme A. It is inhibited by malonyl coenzyme A. CPT1 is inhibited by malonyl coenzyme A. So activation basically means it is bringing in the acyl CoA. It is bringing in the acyl CoA. So this fatty acid is present on the outer side of mitochondria, so it will be brought inside. This is important because beta oxidation takes place only in the mitochondria. Beta oxidation takes place only in the mitochondria. So basically in lipogenesis, palmitic acid was formed. In beta oxidation, palmitic acid will be converted to acetyl CoA. So in beta oxidation, it is conversion of 16 carbon palmitic acid to 8 molecules of acetyl CoA. And these are the steps. None of them is important. Do not worry. None of them is important. You just have to remember here in the end that acetyl CoA is formed. Just that much. That it forms in the end acetyl CoA. So one carbon is, palmitic acid is 16 carbon. Ultimately it forms 8 acetyl CoA. Because acetyl CoA is 2 carbon each. So it is 8 acetyl CoA. However, the energetics is important here. The energetics is important. You just have to remember that one palmitic acid gives 106 ATP. One palmitic acid gives 106 ATP. This is, all of this is important. Now since you believed me so easily कि कोई भी step important नहीं है beta oxidation में, I want you to believe this also कि ये इस slide में हर step important है. So odd chain fatty acid gives rise to propionyl CoA, which is 3 carbon compound, which will be converted to methyl malonyl CoA by the enzyme propionyl CoA carboxylase. This carboxylase is also a biotin dependent carboxylase. What was biotin? Vitamin H. Deficiency of biotin will result in alopecia and dermatitis. Yes, it results in alopecia, and that is why there was a question which states that which of the following will be, which vitamin deficiency should be given in alopecia. So it is of course biotin, and even you can see this for many shampoos. They put biotin in them so that they can spend the same, sell the same 200 shampoo for 2000 rupees. Biotinated shampoos. So that is because biotin decreases the alopecia, or biotin deficiency results in alopecia. Another thing is biotin deficiency is otherwise rare except for people who eat raw eggs. Raw egg has a protein known as avidin, and it is avidin that binds biotin. And hence there was a question that which of the following will increase if biotin is deficient. There will be increase in propionyl CoA, because propionyl CoA is converted to methyl malonyl CoA via a biotin dependent step. This enzyme carboxylase requires biotin. The next is methyl malonyl CoA mutase, and this is a vitamin B12 requiring step, and it will form succinyl CoA. Very important step again, because vitamin B12 is required here. Vitamin B12 is present in no plant source, only animal source have it. So strict vegans are at risk of developing vitamin B12 deficiency. So this here will increase. अगर किसी के पास vitamin B12 नहीं है, क्या बढ़ेगा? Methyl malonyl CoA, and it interferes with myelin sheath formation, and therefore in vitamin B12 deficiency you see something known as subacute combined degeneration, which affects both dorso as well as lateral column. मेगालोब्लास्टिक अनीमिया तो होता ही है दोनों B9 और B12 deficiency में, but subacute combined degeneration is seen only in vitamin B12 deficiency. So this is very important. Why? Because ultimately succinyl CoA forms glucose. Please note, even chain fatty acids, they will form acetyl CoA, and acetyl CoA never forms glucose. It never forms glucose. It never forms glucose. But odd chain fatty acids, they form propionyl CoA, and propionyl CoA will form glucose. So even chain fatty acid, they never form glucose, but odd chain fatty acid, they form glucose. And gluconeogenesis, maybe one of the substrates was propionate. We have written there 4 substrates for gluconeogenesis. We have checked there that one of those substrates was propionate. The next is very important pathway, and that is ketone bodies. Very, very, very important. Another very important topic. The fatty acid is broken down by the process of beta oxidation in liver, and it forms acetoacetate, which is the first ketone body. The acetoacetate will form acetone and beta hydroxybutyrate. These are the 3 ketone bodies: acetoacetate, acetone and beta hydroxybutyrate. Please note, the acetone is volatile and it will not be utilized by any of the tissues, because acetone is so volatile that we exhale it, and hence we say that people who are having diabetic ketoacidosis, they release acetone in breath, and acetone gives characteristic fruity odor to the breath. So this acetone has fruity odor. It gives fruity odor to the breath. But acetoacetate and beta hydroxybutyrate, that can be used for energy. In the extrahepatic tissues, acetoacetate and beta hydroxybutyrate forms acetyl CoA back, and this here will be used for entering TCA, and TCA provides 10 ATP per cycle. So ketone bodies are also small packets of energy, and the formation of ketone bodies occurs in the presence of the hormone glucagon. It occurs in the presence of glucagon. But the most important enzyme here is this CoA transferase called thiophorase. This enzyme is absent in liver. So where are ketone bodies formed? In liver, but they cannot be used by liver because liver does not have the enzyme thiophorase. So liver is the sole producer of ketone bodies but cannot use ketone bodies because it does not have the enzyme thiophorase, and that is one of the most important questions in ketone bodies. Ketone bodies are formed in the presence of the hormone glucagon, and by the beta oxidation of fatty acid, because the breaking will occur in the presence of the hormone glucagon. Lipogenesis will take place in the presence of the hormone insulin. The synthetic, the anabolic processes is caused by the anabolic hormone insulin. The catabolic processes, the breakdown is caused by the hormone glucagon. Cholesterol में note the most important rate limiting enzyme is HMG CoA reductase, and it is inhibited by which drug? Statins, HS drug. We give one drug at night because it causes myalgia. That is the side effect for statins. Small amount of muscle weakness it will cause, statins cause. This step also requires NADPH, which comes from HMP shunt. So not many are important steps here. You just have to remember the rate limiting step. The bile acids. Name. Primary bile acids, cholic and chenodeoxycholic acid. The secondary bile acids are deoxycholic acid and lithocholic acid. Note down the name. Bile acid is the end product of cholesterol metabolism. So cholesterol forms bile acid. Bile acid and bile salt is one and the same thing. If you add sodium to it, it will become bile salt. Otherwise it is bile acid. Right? So in the liver, the primary bile acids are formed, and in the intestine, the secondary bile acids are formed. The primary bile acids are cholic acid, chenodeoxycholic acid. The secondary bile acids are deoxycholic acid and lithocholic acid. Those are your secondary bile acids. The rate limiting enzyme here is 7-alpha hydroxylation of cholesterol by the enzyme 7-alpha hydroxylase. (98 to 99% of this bile acid will go back into the liver via enterohepatic circulation, because bile acid is used for emulsification of fat. It will cause emulsification and helps in the absorption of fat. The lipoproteins is most important topic under lipid. I'll tell you the most important questions here directly. Which is the good cholesterol? HH. This is high density lipoprotein. Which is the bad cholesterol? LDL. The chylomicron is rich in which type of lipid? Exogenous or dietary triacylglycerol? I am covering few points about chylomicron. The chylomicron is synthesized in intestine and is 98 to 99% lipid, while only 1 to 2% protein. And this protein is ApoB48. ApoB48 is unique to chylomicron. Nobody else has it. I am writing all the questions related to chylomicron. The chylomicron gives milky appearance to serum or turbid appearance to serum. And that is why you might have all heard that you should not give a sample after having a hearty meal, because if you will give a sample after having a good meal, it will have chylomicron, and chylomicron will make that serum go turbid, and that is why most of the tests are asked for fasting, so that there is no chylomicron interference. So milky turbidity or chyle-like appearance of serum is due to chylomicron in the serum. The chylomicron is digested by the enzyme LPL, that stands for lipoprotein lipase. This enzyme converts the triacylglycerol to glycerol and 3 free fatty acids. Triacylglycerol. And what type of triacylglycerol it is? Exogenous dietary triacylglycerol. सबसे पहले हमने यही पॉइंट लिखा था, it is dietary triacylglycerol, exogenous triacylglycerol. This here requires a must cofactor, and that is ApoC2, apolipoprotein C2. Similarly, add a note on VLDL. VLDL is synthesized in liver. It has ApoB100 and other apolipoproteins like A1, A2, C1, C2 and small amount of E. And small amount of E. The VLDL is taking the endogenous triacylglycerol. Unlike chylomicron, which was taking the exogenous triacylglycerol, VLDL has endogenous triacylglycerol. That is synthesized by the liver. Now, similarly for LDL, the LDL is rich in cholesterol and has the only apolipoprotein associated with it is B100. And because it is rich in cholesterol, it is highly atherogenic, or it is directly related to coronary artery disease. More is LDL, more are the chances of atherosclerosis, and that is why it is bad cholesterol. The LDL cholesterol is calculated using a formula that says LDL cholesterol is equal to total cholesterol minus HDL and serum triacylglycerol divided by 5. Serum triacylglycerol divided by 5. That is how we calculate LDL. Every time you ask for lipid profile, it is given calculated LDL. They don't estimate the actual LDL in the blood. They calculate using this formula. Of course, the computers are doing it, but it will give you a calculated, not actually estimated LDL. Then we have HDL. HDL is responsible for reverse cholesterol transport. It is having pre-beta HDL, which is a type of HDL and is the most important dyslipidemic marker. higher is pre-beta HDL, lower is the chances of coronary artery disease. It is inversely related. It is inversely related to coronary artery disease. HDL is doing reverse cholesterol transport. That means it is taking cholesterol from the extrahepatic tissue and giving it to liver. And that is why it is considered good. Why? Because जो भी extra cholesterol होगा किसी भी tissue के पास वो HDL को दे देगा और HDL will take it to liver. That means there is no deposition of cholesterol in the extrahepatic tissue. And because there is no deposition, it is inversely related to coronary artery disease, and that is why it has good reputation in market. We like it, we call it good cholesterol. So that was questions on lipoproteins. The cholesterol so these are detailed metabolic pathways of them, but I have given you the most important points under this. Out of hyperlipoproteinemia, the most important lipoproteinemia is hypercholesterolemia type 2. It is defective LDL receptor. The LDL receptor has two portions. One is ApoB100 and other is ApoE. One is ApoB100 and other is ApoE. Out of both, it is having two portion. मतलब ये उसके दो parts हैं LDL receptor के. B100 portion ने जहाँ bind करना था, that is defective, and that is what is asked. That which portion is defective in the LDL receptor? And the answer is ApoB100 portion of the LDL receptor. And that results in, because when we were discussing LDL, we said that LDL के पास सिर्फ और सिर्फ B100 है, तो इसीलिए LDL will not be able to bind and its concentration will increase in blood, and more is LDL in blood, it is associated with hypercholesterolemia and more chances of coronary artery disease. And that is why there is increased risk of coronary artery disease for type 2 hyperlipoproteinemias. So that was all about lipids. The next is amino acids. I'll tell you important points about all the amino acids one by one. All the things which you need to remember, I'll mark it. This is overwhelming. There will be many things here which you have to remember, especially everything about the 3 aromatic amino acids. Those are the most important amino acids, and 3 aromatic amino acids are phenylalanine, tyrosine, tryptophan. तो वो जहाँ जहाँ आएगा वो सबसे important हो ही जाएंगे. So the list of the essential and non-essential amino acid, very important. You can use any mnemonic to remember them, but you must remember the 10 essential amino acids. Which of these are 2 semi-essential amino acids? Histidine and arginine. These are semi-essential. Why? Because it is important for children. Adults can form them, and that is why we call histidine and arginine semi-essential. Essential non-essential तो पता ही होने चाहिए. कौन से 10 essential amino acid है, उनमें से histidine and arginine is semi-essential. Then the biosynthesis of urea. Very important topic. Number 1 is transamination. Every amino acid undergoes transamination. Although 2 are very famous transaminases. One is ALT and another is AST. ALT and AST, and they require vitamin B6. Very important. And there is a vague question which says Vitamin B6 is required for which of the following digestion or metabolism of which of the following compounds? Answer is amino acids or protein. दोनों में से कोई एक ऑप्शन में दिया होगा. There are two transaminases very famous. They have celebrity status. ALT and AST. Why? Why everybody knows about them? They are used as, you also know ALT and AST. नहीं बताना वो बात अलग है. But you also know them. What are they? They are liver function test. Which is more specific for liver function diseases? ALT. Which one will become more specific in alcoholic liver disease? AST. शराबियों में AST बढ़ जाता है, SS. That is how I remember it. So ALT, AST both are liver diseases. They increase in liver diseases. But इसी तरह से हर amino acid undergoes transamination. Only these two we remember because they are required for liver function test. ऐसे ही ग्लाइसिन की भी अपनी ट्रांसएमिनेज है, हर अमीनो एसिड की अपनी ट्रांसएमिनेज है. Then the last step is oxidative deamination. Very important. Oxidative deamination. It converts glutamate directly to alpha ketoglutarate. Together transamination and deamination is called transdeamination. Please note what is the enzyme of oxidative deamination. The enzyme here is glutamate dehydrogenase. This enzyme here is glutamate dehydrogenase. It releases free ammonia. Transamination never releases free ammonia. In transamination, there is no free ammonia released. In deamination, there is release of free ammonia. We are very much afraid of releasing free ammonia because high ammonia in blood can cross blood-brain barrier and cause cerebral edema, coma and death. And that is why we want to translocate ammonia but in non-toxic form. And that is why we convert it to urea and excrete it. So urea is non-toxic carrier of ammonia ion. Another non-toxic carrier of ammonia is glutamine. Glutamine is non-toxic transporter of ammonia ion. It is non-toxic transporter of ammonia ion. And then coming to urea cycle. Which is the rate limiting enzyme for urea cycle? The first enzyme, CPS1. This is rate limiting enzyme. What is the byproduct of urea cycle? Fumarate is the byproduct of urea cycle. Urea is having two nitrogens. One nitrogen comes from ammonia and other nitrogen comes from aspartate. We will revise this quickly. I will tell you the intermediates, you will tell me the enzymes. It's given there in your notes. Carbon dioxide reacts with ammonia to form carbamoyl phosphate. What is the name of the enzyme? Check. CPS1. Can you locate CPS1? Carbamoyl phosphate synthetase 1. This is the rate limiting enzyme. Carbamoyl phosphate reacts with ornithine to form citrulline. What is the name of the enzyme? Check it, it's given in your notes. The second enzyme is ornithine transcarbamylase. The citrulline comes outside into the cytosol. The first two reactions of urea cycle take place in the mitochondria and the last three steps takes place in the cytosol. Please note that the citrulline, once it is outside, it reacts with aspartate to form argininosuccinate. Argininosuccinate releases fumarate and forms arginine, and arginine then releases urea and forms ornithine. So the last three enzymes you will tell me what is the name. Check and tell. Third, fourth and fifth. The third enzyme is argininosuccinate synthetase. Fourth enzyme is argininosuccinate lyase, and the fifth enzyme is arginase. Which enzyme releases urea? Arginase. Arginase. The two nitrogens of urea, one come from ammonia, one come from aspartate. The byproduct of urea cycle is fumarate. So those are most important questions under urea cycle. The most common enzyme defect of urea cycle is OTC, ornithine transcarbamylase, and that is seen as hyperammonemia type 2. Type 2. Also add here that CPS1 requires NAG, N-acetyl glutamate. उसी में दिया ही हुआ है. It requires N-acetyl glutamate as an activator. CPS1. जैसे we were discussing for carboxylases, they require biotin. Similarly, CPS1 here requires NAG, N-acetyl glutamate. In glycine, I want you to remember the specialized products. Purine, glutathione, heme, creatinine, conjugation, and glycine reacts with benzoic acid to form hippuric acid. These are significance of glycine. Creatine, याद रखना, is made of 3 amino acids, that is glycine, arginine and methionine. When creatine is converted to creatine phosphate, if I am adding phosphate, what will be the name of the enzyme? Kinase. Always kinases add phosphate. This kinase is of 3 types. You have CK1, CK2, CK3. CK1 is BB, 2 is MB and 3 is MM. Which one will increase in MI patient? MB. MB is specific to heart, BB is specific to brain, and MM is specific to muscles. And that is why if somebody is having MI, CK2 rises. Somebody is having brain injury, CK1 will rise. Somebody is having rhabdomyolysis, muscle injury, CK3 will rise. The most important topic under glycine is the specialized products of glycine. For branch chain amino acids, I just want you to remember one disorder, and that is maple syrup urine disease. It is a defective alpha keto acid dehydrogenase. This enzyme is an enzyme similar to PDH. It is an enzyme similar to PDH, and under PDH we have written that it requires tender love care for Nancy. है ना? What was this? Thymine, lipoic acid, coenzyme A, FAD and NAD. The same 5 cofactor is required by this branch chain keto acid dehydrogenase. And if this enzyme is defective, it results in maple syrup urine disease. The branch chain amino acids is catabolized using a common enzyme called branch chain keto acid dehydrogenase. If this enzyme is defective, it will result in accumulation of the branch chain keto acids, valine, leucine, isoleucine, and hence the disorder is called, it gives a characteristics sweetish odor to the urine or burnt sugar like odor to the urine, and hence the disorder is called maple syrup urine disease. अच्छा, another thing is, in this characteristic finding क्या है कि बच्चा जो है at birth is completely normal, but after first 15 days of life the child starts developing characteristic opisthotonous posture, that is hyper extended body, and then he will at times become opisthotonous, then flaccid, then opisthotonous, then flaccid. And because of this alternate muscle involvement, the child develops extreme muscle weakness. So if you see the term opisthotonous or hyper extended body, it is seen only in maple syrup urine disease, characteristic to maple syrup urine disease, right? Then everything about phenylalanine and tyrosine and tryptophan is important. Everything, word to word, everything is important. So this is the most important amino acids. The phenylalanine is converted to tyrosine by an enzyme known as phenylalanine alpha hydroxylase. If this enzyme is defective, it results in a disorder known as phenylketonuria. This enzyme requires tetrahydrobiopterin, and tetrahydrobiopterin requires an enzyme dihydrobiopterin reductase. It will convert dihydrobiopterin to tetrahydrobiopterin. Everything is important here. Phenylalanine alpha hydroxylase is the most common cause of phenylketonuria. However, if somebody does not have tetrahydrobiopterin या फिर dihydrobiopterin reductase is defective, still it will result in phenylketonuria. So the classical cause is defective phenyl alpha hydroxylase, phenylalanine alpha hydroxylase. Classical in biochemistry means most common. So the most common is defective phenylalanine alpha hydroxylase. The specialized products of tyrosine, all of them are important. All of them are important. Melanin, catecholamines. What are the catecholamines? Dopa, dopamine, norepinephrine and epinephrine. Tyramine, everything here is important. This pathway, each step is important. Tyrosine to dopa, the enzyme is tyrosine hydroxylase. This step also requires tetrahydrobiopterin. Dopa to dopamine, the enzyme is dopa decarboxylase. Dopamine to norepinephrine, the enzyme is dopamine oxidase or dopamine hydroxylase. This carboxylase requires vitamin B6. Decarboxylase requires vitamin B6. For any reaction where there is decarboxylase, it will require vitamin B6. And the dopamine hydroxylase or oxidase requires vitamin C. The only difference between norepinephrine and epinephrine is of methyl group, and that methyl group comes from methionine. It comes from methionine. So the last enzyme is a methyl transferase. Every step here is important. And every disorder related to tyrosine, phenylalanine, tryptophan, very important. Phenylketonuria, yes. What is alkaptonuria? Deficiency of the enzyme homogenetisic acid oxidase. As a result, what will happen? Homogenetisic acid will increase, and this on oxidation it causes benzoquinone acetate. And it is this benzoquinone acetate which is black in color. And that is why it gives a characteristic black colored urine. It also causes blackening of cartilage, and that is known as ochronosis. Ochronosis is blackening of cartilage. Then we have tyrosinemia, albinism. It is all due to related to tyrosine metabolism. Tyrosinemia type 1, is fumaryl acetoacetate hydroxylase defect, and deficiency of this enzyme causes boiled cabbage like udder to urine, and same is for tyrosinemia type 2. Type 2 is defective tyrosine transaminase. This also is associated with boiled cabbage like udder to urine. Boiled cabbage like udder to urine. Tyrosine transaminase defect. So tyrosinemia type 1 and type 2, very poor prognosis disease. They are associated with liver cancer. The next important amino acid is tryptophan. Everything related to tryptophan is important. It requires forms melatonin, it forms serotonin, and it is characteristically metabolized via kynureninase enzyme. This enzyme requires vitamin B6, pyridoxal phosphate. And of course the Hartnupp's disease. The tryptophan forms niacin. It forms niacin, and niacin is a vitamin B3, and hence niacin is also known as endogenous vitamin. It is known as endogenous vitamin. And deficiency of niacin is seen as pellagra is characterized by 4 D's: diarrhea, dermatitis, characteristic type of dermatitis seen in neck region known as Cassell's necklace, dementia and death. In methionine is much more important as compared to cysteine, the sulfur containing amino acid. And here the enzyme cystathionine beta synthase is very important. They have also highlighted this in the diagram. It is in the flow chart. Cystathionine beta synthase requires vitamin B6, and deficiency of both cystathionine and vitamin B6 will result in homocystinemia, high levels of homocysteine. The homocysteine is also converted to methionine via methyl B12. The only difference between homocysteine and methionine is of B12, of methionine, and that comes from, the methyl group is coming from vitamin B12. This enzyme here is called homocysteine methyltransferase. If somebody does not have the enzyme homocysteine methyltransferase or methyl B12, it will also result in high homocysteine. So those are also causes of homocystinemia. So all of them are causes of homocystinemia: methyl B12 deficiency, B6 deficiency, cystathionine beta synthase deficiency, and homocysteine methyltransferase. High homocysteine is directly related to coronary artery disease or any thromboembolic disorder like stroke, DVT, pulmonary embolism. In collagen, I'll point at the most important step, and that is conversion of proline to hydroxyproline. In collagen, wherever proline is present, 33% of collagen is glycine, and at 10% of locations, the proline, wherever it is present, it is converted to hydroxyproline. This enzyme here is prolyl hydroxylase, and this enzyme requires vitamin C and iron, and that is why this hydroxyproline is required for the hydrogen bond formation, and that is why if somebody does not have vitamin C, collagen formation is defective, and that is seen as scurvy, easy bruisability of gums, because collagen is required for the vasculature, blood vessels. Collagen is the most abundant tissue in our body, it forms blood vessels. And most abundant plasma protein in our body is albumin. The most abundant protein is collagen, and collagen form blood vessels, and उसके लिए hydrogen bonds बहुत ज़रूरी है for the stability of collagen, and that is why vitamin C deficiency will result in poor hydrogen bond formation or poor collagen formation, and that is why it results in easy bruisability seen in scurvy. This kind of modification is called post-translational modification. It is most important post-translational modification requiring vitamin C. Our next topic is vitamins. Very, very, very important topic. The fat soluble vitamins is ADEK and ascorbic acid and your B complex. Those two are your water soluble vitamins, but B complex itself has many other vitamins in it. The active form of vitamin A is all of them. Retinol, retinal, retinoic acid, they are all active form. Keep on writing along with me that these three are active forms of vitamin A. You'll find questions like which of the following is active form of vitamin A? They are all. One of the options will be all. Then function of vitamin A, important in vision, growth and differentiation. It has antioxidant properties. Three vitamins, they have antioxidant properties, and these are vitamin A, C and E. Which is most important antioxidant? Vitamin E. Deficiency causes first symptom night blindness, nyctalopia. The confirmatory symptom is Bitot spot. It can result in dryness of conjunctiva, dryness of cornea, ultimately resulting in keratomalacia. Keratomalacia is liquefaction of cornea. Once this has occurred, it will become irreversible damage, and that is why vitamin A deficiency is most common cause of preventable blindness worldwide. Then when revising, go through all vitamins one by one. Whatever is given in this notes, right, just revise whole of it one by one. I will be pinpointing the most important points only, right. Vitamins mein do not skip any line here, right. Everything is important while you are revising it. I am just pointing out the most important points. Vitamin D has steroid hormone like action and is formed from 7-dehydrocholesterol, very important pathway. 7-dehydrocholesterol ultimately forms vitamin 24, 25 and 125 vitamin D3, which is the active form of vitamin D3. 125 is the active form. Which enzyme is present in the liver? 25 alpha hydroxylase. The next two enzymes are present in kidneys, 24 and 1-alpha hydroxylase. These are enzymes of kidneys. It's given in the text. I have marked here, but it's given in the text supporting that diagram. Function, calcium homeostasis. Which other instead of vitamin, which other hormones are required for calcium homeostasis? Parathyroid hormone and calcitonin. Do not skip any of these points. Vitamin E, just remember it is most potent antioxidant, tocopherol. Vitamin K, this part, very, very, very important. During this reaction, note that factor 2, 7, 9, 10 are carboxylated and they will form carboxylated glutamic acid. During this reaction, we require vitamin K, and this will form active form of factor 2, 7, 9 and 10. So, the carboxylated glutamic acid, this one here is carboxylated glutamic acid, is called GLA, G-L-A. It is part of the active 2, 7, 9, 10. It is not seen in, glutamic acid is seen in 2, 7, 9, 10, but carboxylated glutamic acid is an example of post-translational modification. Post-translational modification. Like we saw for vitamin C, similarly there is a vitamin K dependent post-translational modification. During this step, hydroxyquinone form of vitamin K forms epoxide form of vitamin K, and we have to convert it back. For this we require an enzyme called epoxide reductase. This enzyme is inhibited by a drug known as warfarin. And hence warfarin is an anticoagulant drug. Water soluble vitamins, everything is important. I'll tell you just the function and deficiency. B1 is required for PDH transketolase, most important. Deficiency of B1 in alcoholics result in, yes, Wernicke-Korsakoff psychosis. In non-alcoholics it will result in beriberi. Riboflavin, it is also required by anywhere you have written FMN, FAD. Then all these are FMN, FAD containing compounds requiring reactions. Deficiency of vitamin B2 is stomatitis, angular stomatitis. It is called inflammation at the angles of lips, and that is known as cheilosis. It causes glossitis, it causes seborrhea, dryness of scalp and photophobia. So non-specific signs and symptoms. Niacin deficiency already discussed, pellagra. Coenzyme A deficiency, it will cause exfoliative dermatitis. B7 deficiency, alopecia. And B6 is very, very, very important. This one here. Out of all the water soluble B complex deficiencies, B6, B9, B12, most important ones, and of course B1, B6, B9, B12, these are the same one which are required in alcoholics. So these are the most important ones. B9 deficiency. We give B9 supplements to pregnant female, right? And because deficiency can cause neural tube defects. This is required for 1-carbon metabolism. B9 is required for 1-carbon metabolism. B9 deficiency, I have few questions in the end. I'll be discussing B9 and B12 pertaining to those questions in the end again. So these, that is why I've just skipped those topics. But when you are revising, I want you to revise word to word of all this B complex as well as for all the vitamins. I'll be discussing whatever questions are related to them in the end. So I've just gone quickly through all these vitamins. The next topic is enzymes. Under enzymes, I'll tell you again the most important one. What are coenzymes? What are coenzymes? Coenzymes are also called co-substrate. They are also called co-substrate. Any vitamin which is loosely bound to the enzyme is called coenzyme. Similarly, what is holoenzyme? Some terminologies. Holoenzyme means complete enzyme. What is apoenzyme? Apoenzyme means only protein part of the enzyme. Coenzyme means vitamins required by the enzyme. So similar terminologies can be confusing. Holoenzyme, complete enzyme. Apoenzyme, only protein part of the enzyme. You can skip this specificity. You can skip the properties of enzymes. Factors affecting is important. Here I want you to remember what is Km. Km is equal to the substrate concentration at which initial velocity is half of Vmax. You must remember the definition for Km. Km is that substrate concentration at which initial velocity is half of Vmax. Higher is the Km, lower is the affinity. Lower is the Km, higher is the affinity. We saw this for GLUT2 and GLUT4. The next slide. I'll give you a diagram here, and this slide is the most important slide under enzymes. What do inhibitors do to the Line-Weaver-Burke plot? Let's see. There are two types of inhibitors, competitive and non-competitive enzyme inhibitors. If this is a reaction in the absence of an inhibitor, the same reaction in the presence of a competitive inhibitor will behave like this. For non-competitive inhibitor, if this is a reaction in the absence of a non-competitive inhibitor, the same reaction will look like this. So this is in the presence of a non-competitive enzyme inhibitor. That means Km has increased, Vmax is same for competitive enzyme inhibitor. Km is same for non-competitive because the starting point is same, and Vmax will decrease. So you have to remember this. If you see a cross in the examination, it is competitive. If you see a V in the graph, it is victory, no competition, right? So Km is same for the non-competitive, Vmax will decrease. This is the most important slide in these enzymes. Then Km increases and Vmax remains same, that is for competitive inhibitor. Km increase, Vmax same, that is competition, and Km same, Vmax decreases, there is no competition. The inhibitor does not resemble the substrate. You will not be asked the mechanism. ये कैसे आया? Nobody will ask you. They'll just ask you this. So इसको रट्टा लगा लो and this put this under last minute revision topic. Right? Because again you will forget this. What will happen to Km and Vmax? That is not what I wish, but that will happen inevitably. So this is the most important topic under the enzymes. In heme, what is the rate limiting enzyme? Check here. Under the pathway, it is the alright, write down here only. Just 2-3 important points. ALA synthase requires vitamin B6, is the rate limiting enzyme for heme biosynthesis and is inhibited by the end product, and that is heme. If somebody does not have ALA synthase or is having vitamin B6 deficiency, it will result in sideroblastosis, sideroblastic anemia, ring shaped RBCs, characteristic to the heme deficient anemia or deficiency of ALA synthase or vitamin B6. This here ALA synthase is given. Any of the enzyme for ALA synthase of the heme synthesis, if defective, can result in porphyria. Any of the enzyme, if defective, can result in porphyria. For porphyrias, remember just two major porphyrias. Number one is acquired intermittent porphyria. And number two is porphyria cutanea tarda. Only these two porphyrias I want you to remember. In acquired intermittent porphyria, the enzyme that is defective is UPG1 synthase. In porphyria cutanea tarda, the enzyme that is defective is UPG3 decarboxylase. In acquired intermittent porphyria, as the name is suggesting, it is intermittent. These people, they are normal for the rest of their lives, but one fine day they will develop acute intermittent porphyria. That means acutely they will start developing abdominal pain, right? So they present with either abdominal pain or neuropsychiatric symptoms. And in porphyria cutanea tarda, as the name suggests, it is easier to remember. Porphyria cutanea tarda mein, it is cutaneous manifestation. These people, they have blistering of skin, extreme form of blistering of skin all over the sun exposed areas in the body. So they develop blistering of skin. Extreme hyperpigmentation. Ultimately blistering will get sheared and it results in extreme type of hyperpigmentation. Only 2 porphyrias you have to remember, and their enzyme defects. UPG1 synthase defect will result in acquired or acute intermittent porphyria, and porphyria cutanea tarda is defective UPG3 decarboxylase. Both have different symptoms. In intermittent porphyria, these people still exist, जिनके abdomen में you will find multiple laparoscopic scars, because every time they came with acute abdominal pain, they were explored for either appendicitis or pancreatitis or for cholecystitis. So these people, they still exist who have this kind of intermittent disorder. They have normal episodes of life, and then suddenly one day they will develop these symptoms. HbA1c itself is an important topic. Glycosylated HbA1c normally should be less than 6.5 gram %, but if it is more than that, it is pointing towards diabetes mellitus, and this is non-enzymatic binding of hemoglobin to glucose. It is when the glucose is very high in the blood, it just binds with hemoglobin. The last topic is electron transport chain. I'll explain this. Most of the students have a very wrong concept for electron transport chain. Let's see. The electron transport chain is present in the inner mitochondrial membrane. In the inner mitochondrial membrane, there is complex 1, complex 2 here, complex 3 here, and complex 4. This side is the matrix side, and this side is the intermembrane space. So you have mitochondrial matrix towards the lower side, and above is the space between the two membranes. Mitochondria is a double layered organelle. The donor of electron to the complex 1 is NADH. The donor of electron to the complex 2 is FADH2. These complexes large proteins. So what they will do is they will transport it to ubiquinone. Ubiquinone is a mobile segment. Ubiquinone is also called coenzyme Q or Q10. This ubiquinone gives the electron to complex 3. Complex 3 again is a protein, cannot move. It will give to another mobile complex called cytochrome small c. Cytochrome small c will give the electron to complex 4, and complex 4 will give the electron to final electron acceptor, which is oxygen. And oxygen during this reaction is converted to water. There is one of the questions related to this. There are many questions related to electron transport chain, and hence I am explaining it. So the complexes are fixed complexes. They cannot move, but they can take electron from NADH and FADH2. Once they take the electron from them, they will pass it to next. Electron always moves from 1 to 3 to 4 or 2 to 3 to 4. Is this clear? Why? Because you might have all noticed that battery में भी एक side positive लिखा होता है और एक side negative लिखा होता है. Inside the battery, the electron is moving from negative ion to the positive electrode, right? The same reason is here. The NADH is minus 0.32 volts and oxygen is plus 0.80 volts, and that is why electron will always move from its higher concentration to a region of its lower concentration, forming a type of electric current. When this is happening, simultaneously the protons, they cross the complex 1, 3 and 4. 4 protons cross complex 1, 2 protons cross complex 3 and 4 protons cross complex 4. This is fixed for complex 1. For 3 and 4 it is not fixed. Some authors believe it is 4-4-2, others believe it is 4-4. But they all believe that complex 1 is transporting only 4 protons. And the total amount of protons transported by one NADH is 10 protons. Ultimately, these protons must come back inside. And when they come back inside, they come back through FOF1 subunit. When this FOF1 subunit is activated, simultaneously ADP will be converted to ATP. And this is the major oxidative phosphorylation. What you are now seeing in front of you are the two processes which are coupled processes. One is electron transport chain which has created proton gradient, and that proton gradient has formed ATP. Have you seen dam? Not dams, dam. Where the water is stored at a very high height. One day we will lower the gates. The water will fall on turbine. The turbine will get rotated, and that turbine rotation will convert the mechanical energy to light energy. The same way it is happening here. Here also you are converting the proton gradient, which is a type of potential energy, to a smallest motor in the FO subunit that gets rotated. And once that is rotated, it will convert ADP to ATP. So it is transfer of energy from one form to another. The way the first law of thermodynamics stays. Energy can neither be produced nor be destroyed. You are just changing it from one form to another. So here we are changing proton gradient to ATP. I repeat, there are two processes, electron transport chain and oxidative phosphorylation. Both are coupled process. अगर इलेक्ट्रॉन ट्रांसपोर्ट चेन होगा तो ATP synthesis होगा ही होगा. Unless, let's say I give a drug which has created a pore in the membrane. It has created a pore in the membrane. This will allow easy passage of these protons. अब वो पोर से अंदर आराम से आ जाएंगे. Will there be ATP synthesis? No, there is no ATP synthesis. For ATP synthesis to occur, hydrogen must come back through FOF1 subunit. So there is no ATP synthesis. Will there be electron transport chain? Yes, ETC will occur because ETC का requirement क्या है? Protons चाहिए उसको. Protons तो अंदर आ गए ना, तो अब वो फिर से protons को बाहर circulate करता रहेगा. So the two processes which were otherwise coupled is now uncoupled, and these compounds which create pores in the membranes are called uncouplers. They broke the marriage between electron transport chain and oxidative phosphorylation. The most classical example of uncoupler is thermogenin, which is present in brown adipose tissue, which is found in the nape of neck region of the newborn. And if otherwise, अगर uncouplers को छोड़ दो, otherwise the two processes are always coupled. अगर एक होगा तो दूसरा होगा ही होगा. सिर्फ uncouplers के केस में ऐसा नहीं होता है. So if now the next important topic under this is inhibitors. And here, this is the whole list of inhibitors in front of you. I want you to mainly remember the inhibitor for complex 2 is malonate, and the inhibitor for complex 4 is hydrogen sulfide, carbon monoxide and cyanide. If complexes are inhibited, ultimately your ATP synthesis will also get inhibited. Now, if NADH is the starting substrate, how many protons are formed? How many protons traverse the mitochondrial membrane? Yes, 10 protons. अगर NADH starting है तो 10 total protons, and that is why it is forming 2.5 ATP. If FADH2 is the starting substrate, it will transport how many protons? Only 6 protons. And that is why we say that FADH2 forms less energy, because it is transporting less protons, right? It is forming only 1.5 ATP. You will use newer calculation. According to newer calculation, one NADH is 2.5 ATP and one FADH2 is 1.5 ATP. You still have some older books, some older questions where they have calculated using 3 ATP for NADH and 2 ATP for FADH2, which is no more the norm now, right? So that was all of your quick revision, and we will now discuss the questions related to it. The next half an hour to 45 minutes will be all your latest questions last 4 to 5 years. Question number 1. In fatty liver which is not given, what is your answer? Here only, in the notes also it is given something known as lipotropic factors. The lipotropic factors are those factors which prevent fatty liver formation. These are, write down a mnemonic that says choline, lecithin, inositol, methionine and betaine. The mnemonic is CLIMB. CLIMB. These are those factors which are required to prevent fatty liver formation. Choline, lecithin, inositol, methionine, betaine. Out of which, in the options given here, choline and methionine is given, right? In addition, all the vitamins and antioxidants, they also will prevent fatty liver. That is from our understanding of common sense, that you should take vitamin supplementations, and right, it will prevent fatty liver formation. So that is B9 is also, and anyhow ethanol should not be given unless somebody is having methanol poisoning, right? So ethanol is not a part of the, it will rather cause fatty liver formation. That is what happens in alcoholic liver diseases. Ethanol forms acetate, and acetate is converted to fatty acid, palmitic acid. And that is why ethanol is associated with fatty liver formation, right? So the answer here of course is ethanol. Question number 2. In a disease inheritance, mother side has a disease, her son also has same disease. Identify the type of inheritance. It is mother is affected, so the X chromosome is affected and it is getting transmitted to only son. So it is X-linked recessive. A patient has swelling in the metacarpophalangeal joints. Serum uric acid levels are raised. Doctor prescribed a medicine for the same. The medicine will act by inhibiting. What drug is this? Allopurinol. Allopurinol is converted to alloxanthine, and this will be converted to, this combines with the enzyme xanthine oxidase. It combines with the enzyme xanthine oxidase in such a way that it will never leave the binding site of the enzyme. So product to kabhi nahi banega, because it is never going to leave the binding site of the enzyme. But at the same time, xanthine oxidase is inactivated because it is never going to release from its binding site. So what was the normal function of this enzyme xanthine oxidase? It was going to convert xanthine to uric acid. And xanthine is formed from purines. And what are the purines? Adenine and guanine. So adenine and guanine, ultimately they form xanthine, and xanthine forms uric acid. And hence in us, uric acid is excreted from by the enzyme formation of conversion of xanthine to uric acid. So purine is excretable form is uric acid. So this enzyme xanthine oxidase is forming uric acid, and that is why in the question they have given swelling of metacarpophalangeal joints, or it can be swelling of the any big joint involvement, but usually it is a monoarticular type of joint involvement seen in gouty arthritis. So in gout it is a result of high circulating uric acid over a period of time, and that is why it will result in gouty arthritis. Nitric oxide is synthesized from arginine. Arginine forms nitric oxide, it also forms urea, and it forms keratin, and it forms creatinine. So these are the products of arginine. Arginine also in addition forms histone. Note, histone does not have histidine. There is no histidine in histone. Histone is rich in arginine and lysine, but does not have histidine. Histone is a basic protein. It is rich in arginine and lysine, but does not have histidine. Which of the following is involved in protein folding? Chaperones are themselves proteins that helps in the other proteins to be folded. Proline and arginine तो कहीं से भी नहीं होगा. अब ubiquitin can be a difficult option. Ubiquitin is required for protein degradation. Chaperones help in protein folding, and ubiquitin itself is a protein, but it helps in protein degradation. Along with ubiquitin, remember proteasome. One of the smallest. So this is chaperones, they assist other proteins to fold, और वो खुद भी proteins हैं. Ubiquitin, it helps in other proteins to degrade, but वो खुद भी proteins हैं. Similarly, proteasome is a large protein which is helping other proteins to degrade, but वो खुद भी protein है. So they are all proteins, but they are helping other proteins either to fold or to degrade. For degradation, you just have to remember two proteins. One is ubiquitin, other is proteasome. In alcoholic patient presents with neurological symptoms, there is deficiency of vitamin. Yes, we have discussed this. A female presents with macrocytic anemia, homocystinuria, and normal methyl malonate levels. Which vitamin deficiency can this be? Yes. But why folate plus vitamin B12? मिथाइल मैलोनाइल को तो नॉर्मल है ना इस गिवन क्वेश्चन में. अगर ओनली फोलेट, इसमें ओनली फोलेट आएगा. एक्चुअली दिस इज अ क्वेश्चन सिमिलर टू दिस क्वेश्चन इज ऑल्सो प्रेजेंट. इट इज एन ओल्डर क्वेश्चन. उस क्वेश्चन में था कि पर्सन इज हैविंग मेगैलोब्लास्टिक अनीमिया, what vitamin supplementations you should give to this. One of the options was B9 and B12. उसमें correct answer is B9 and B12. But यहाँ पे because उसमें दिया है methyl malonate, in the discussion we saw methyl malonyl CoA mutase requires only vitamin B12, and if it is normal that means there is no B12 deficiency. अगर methyl malonate CoA deficient नहीं है तभी तो वो normal है ना, B12 normal है, तो इसका मतलब only B9 supplementation is required. So answer is folate only. Question number 8. A heart patient suddenly has a chest pain. Which isoenzyme? Like we saw the isoenzymes for creatine kinase, LDH also has 5 isoenzymes. 1, 2, 3, 4 and 5. LDH2 is normally more than 1 in healthy population. But if somebody is having MI, in them LDH1 will become more than 2. This is called reversal of LDH ratio or flipped ratio. It is seen in cases of MI. LDH1 क्योंकि ज़्यादा present होता है in heart as compared to other tissues, and that is why MI is releasing more LDH1 in blood. So the answer is LDH1. Cofactor for mitochondrial superoxide dismutase is zinc. Actually, this enzyme superoxide dismutase are of 3 types. Zinc, then there is copper, and there is manganese. If all of them are given in the options, like in this case, you'll go with zinc. But most likely this question was all except. Right? इतना closely knit question नहीं देंगे. It will be all except. Menkes kinky hair disease is due to deficiency of copper or copper toxicity or deficiency. It is copper excess. A patient came to OPD with complaints of diarrhea, dermatitis, dementia. It is found to be maize eater. Maize is naturally deficient in tryptophan, and we saw earlier that tryptophan was required to form niacin. So it is niacin deficiency. They can also give an image based question with somebody at neck region having a very large dermatitis known as Castle's necklace. Now, like this is an image based question here. A 40-year-old female came to the OPD with the feature of anemia. She had undergone subtotal gastrectomy few months back. General examination showed following changes in the tongue as shown in the image. Which of the following vitamin is responsible for this? It is glossitis, which is seen. Yes, one of the causes is also B12. One of the causes for glossitis is also vitamin B2. But why am I choosing vitamin B12 over vitamin B2 here? Because it is telling you about the subtotal gastrectomy. For the absorption of vitamin B12, it combines with something known as intrinsic factor. This intrinsic factor comes from the gastric cells or parietal cells. This combines and then together it is absorbed at terminal ileum. And if somebody is having ileal resectomy, in them vitamin B12 can be deficient. Then question number 13. Which of the following condition is true about fed state? सबसे पहले ये बताओ which is the hormone of the fed state, insulin or glucagon? Fed state, it is yes, insulin. And what is the hormone of fasting? Yes, it is glucagon. So glucagon will cause anything which will raise the blood glucose. Insulin, what it causes is, it tends to decrease the blood glucose. Right, and that is why it promotes glycolysis. It increases the activity of the enzyme PFK1 and it increases glycolysis, and that is consumption of glucose. Okay? So which of the following condition is true about fed state? Increase in insulin and decrease in glucagon. That is your answer. Question number 14. Patient who is chronic alcoholic has signs of B1. Which of the following is the marker? RBC transketolase. Acha, RBC glutathione reductase is used for B2. We discussed that earlier. B2 is for glutathione reductase. इसमें options में नहीं दिया है वो. Question 15. A 70-year-old male has bruising under the eyes. Which of the following vitamin deficiency leads to easy bruisability and bleeding gums? Yes, that is what I wanted to emphasize, that ऐसा नहीं है कि सिर्फ gums ही bleed करेंगे. It can be associated with any type of hemorrhage. The only finding in vitamin C deficiency is increased clotting time, increased bleeding time, and it is associated with poor wound healing. There are two vitamins which are associated with poor wound healing. One is vitamin A and other is vitamin C. Both are associated with poor wound healing. And easy bruisability कहीं की भी हो सकती है. It's not only that gums will get bleeding. And because it is a bleeding disorder, this can also present with anemia. Cholesterol is synthesized from acetyl coenzyme A. The acetyl CoA synthesizes cholesterol, but cholesterol is degraded to bile acids. Bile acid, primary bile acid, secondary bile acids. Glucagon. We already discussed glucagon is a hormone of fasting. It is a hormone of fasting. So it will promote anything which will cause catabolic, right? For example, gluconeogenesis. It will promote gluconeogenesis because it will raise blood glucose. Insulin decreases blood glucose, glucagon tends to raise. Then lipolysis. Lipolysis means beta oxidation of fatty acid. What will glucagon do to beta oxidation? Promote it. What will glucagon do to beta oxidation? अगर beta oxidation ज़्यादा होगा तो ketone body synthesis will also be more, right? So ketone body synthesis also increases. We saw it under ketone body synthesis that the acetyl CoA is coming from beta oxidation. So more is beta oxidation, more is ketone body synthesis. Glycolysis is promoted by insulin. So that here is your answer. So glucagon activates all the pathways except glycolysis. Thermogenin is present in mitochondria. The role of thermogenin is also called UCP1, uncoupler protein 1. It is present in brown adipose tissue in the nape of neck region of the newborn, and it prevents them from hypothermia. What is the enzyme defect in Gaucher's? Beta glucocerebrosidase. अच्छा अब ये बताओ, the option B, glucose 6-phosphate, is it an enzyme? No, it is just given there to confuse you कि अगर glucose 6-phosphate भी कहीं याद रह गया तो वो confusion करेगा. What is glucose 6-phosphatase enzyme defect? It is von Goerk's disease. And the last enzyme is your beta glucocerebrosidase, or simply they have mentioned it here as glucocerebrosidase. Which enzyme defect is beta galactocerebrosidase, which we have written as beta galactosidase? Krebs. Alpha galactosidase is, whether you call it alpha galactosidase or alpha galactocerebrosidase, it's one and the same thing, because all these sphingolipids are found in brain. So we call them cerebrosidases. Acha, which enzyme defect is alpha galactocerebrosidase? Fabry's is the only X-linked disease. All the others are autosomal. Fat sugar baby is seen in kwashiorkor. Protein energy malnutrition. There are two types of it. One is kwashiorkor and another is marasmus. In marasmus, it is only energy deficiency. In kwashiorkor, it is both protein as well as energy deficiency. So here it is only energy deficiency. 21. A vegetarian patient presents with anemia and hypersegmented neutrophils. Hmm. Hypersegmented neutrophils are characteristic to vitamin B12 deficiency. So it will result in megaloblastic anemia. Sideroblastosis is B6 deficiency. Which of the following explains the role of vitamin D? Hmm? White sunlight converts 7D hydrocholesterol to cholecalciferol. You can check under that. A तो is 25 alpha hydroxylase. मैंने वहाँ मार्क भी किया था. It is present in liver. So sunlight is a photolysis. The last enzyme here, the last reaction is a photolytic step, and that is done by sunlight. Which enzyme deficiency is most likely to cause classical galactosemia? I have marked there also. It is galactose 1-phosphate uridyl transferase defect. The subcellular organelle which is associated with killing viruses and bacteria is lysosomes, the suicidal bag. Right, it will cause lysis of everything. Which of the following has low glycemic index? Sweet corn. Sweet corn has the lowest. Yes, it has lowest. Even ice cream has very low glycemic index, around 60. Potato has a very high glycemic index. Papaya, watermelon, they are very high glycemic index food. What is severe combined immunodeficiency? ADA, adenosine deaminase defect. It somehow affects only B and T cells, and because it affects by a mechanism not clearly understood, but this enzyme is only deficient in B and T cells, and therefore it results in both loss of humoral activity as immunity and cell mediated immunity. Question number 27. Which of the following is considered as universal methyl donor? S-adenosyl methionine, SAM. You can check under conversion of norepinephrine to epinephrine, where all the reactions they use methyl donor is SAM. Which vitamin deficiency will result in cheilosis? Yes. A patient with intellectual disability was found to have hyperuricemia. Characteristic self-mutilation behavior was noticed. What is the following enzyme defect? It is HGPRTase deficiency. HG stands for hypoxanthine guanine phosphoribosyl transferase. It is this defect which is seen as Lesch-Nyhan syndrome, a characteristic self-mutilation behavior. It is a compulsive biting disorder where the child keeps on biting his own fingers, lips. They are keeping on bleeding. Of course, this child is mentally retarded. A very poor prognosis, X-linked disorder. Which of the following amino acid is excreted in urine in maple syrup urine disease? Branch chain amino acids, and the 3 branch chain amino acids is valine, leucine, isoleucine. So the answer is leucine. Maple syrup urine disease, it was branch chain keto acid dehydrogenase defect, right? Hormones that regulate serum calcium is, I guess we all agree, A, calcitonin, vitamin D and parathyroid hormone. We discussed this earlier. Question number 32. Patient has macrocytic anemia. That is why I said B1, B6, B9, B12. Repeated questions have been asked. Very, very, very important. 4 vitamins. A patient has macrocytic anemia, high homocysteine levels. His methyl malonyl CoA levels are normal. So ये नहीं हो सकता. Macrocytic anemia है तो B9 तो होगा ही, अगर B12 नहीं है. So the answer is folate. Glutathione peroxidase contains which metal? I told you ये confuse करेगा in the examination. The answer is Selenium. We have marked it there. It is selenium. A patient having fatigue and weakness on examination, his hemoglobin was low, iron low, patient that his wound healing is delayed. Which vitamin deficiency? Both अगर दोनों दिया है तो फिर which what is your answer? Why are you going with vitamin B12? Poor wound healing was associated with which two vitamins? A and C. पर A and C दोनों ही options में है. Now we will go with C. C is better answer as compared to A. Fats are stored in which of the following forms in the adipose tissue? It is stored in the form of triacyl glycerol. Triacyl glycerol is the neutral fat. What is the cause of xeroderma pigmentosa? Let's see. In nucleotide excision repair, what happens is, suppose this is DNA. Agar DNA one arm has thymine, what will be present on the opposite side? Adenine. The thymine binds to adenine on the opposite side. But when UV light falls, the thymine, instead of binding to the adenine on the opposite side, they form thymine dimers amongst themselves. This bulky adduct has to be removed, and for this removal we need the enzymes like XP enzymes, xeroderma pigmentosa, and defective XP enzymes is called xeroderma pigmentosa. It is a very poor prognosis disease characterized by early skin cancers, and it causes death by third decade of life because of accumulating thymine dimers. It causes skin cancer. So the answer is nucleotide excision repair. For base excision repair, add here only it is associated with lung cancer, breast cancer. For mismatch repair, it is, if this is defective, it causes hereditary non-polyposis colon cancer. Homologous recombination can result in any cancers. HNPC stands for hereditary non-polyposis colon cancer, also called Lynch syndrome. That is because of defective mismatch repair. We'll study that again in surgery also. Which is the enzyme deficient in Tay-Sachs? Hexosaminidase A. Enzyme defect in von Gerk's? Yes. Do not confuse glucose 6-phosphatase with G6PD. What is this? Yes. A child presents with complaints of urine turning black. Alkaptonuria. What is the enzyme defect? Homogenetisic acid oxidase. Complex 4 is inhibited by? Yes. Both are given. No, it's only cyanide. I thought carbon monoxide is given. Okay. Cyanide. A patient has swelling in metacarpophalangeal joint. I guess this is a repeat, and it is xanthine oxidase. Vitamin deficiency causes tingling sensation. B6. I'll emphasize one important point. B6 deficiency or toxicity, both will appear as tingling sensation, right? It can be associated with vitamin B12 deficiency also, but it has been most commonly found with vitamin B6 deficiency. Another thing is, this deficiency is otherwise rare, but is commonly seen in people who are taking isoniazid, which is part of anti-tubercular treatment regime, and this causes deficiency of vitamin B6. Question number 44. Which of the following enzyme deficiency is likely to cause von Gerk's? Glucose 6-phosphatase. Arginine. Question number 46. A patient was presented to OPD with Features of collagen vascular disorder. Which of the following is likely associated with defective collagen formation? Ascorbic acid and it is collagen defect. So your answer is ascorbic acid and copper. Copper is actually not required by prolyl hydroxylase. We saw it. There is another enzyme known as lysyl oxidase, and this enzyme requires copper, and that enzyme is also required for collagen formation, and hence the answer is B. Which of the following enzymes simultaneously incorporates molecular oxygen and produces water? It is complex 4. Complex 4 is also known as cytochrome oxidase, and hence it is, the answer is cytochrome C oxidase. It's complex 4. It is the technical name for complex 4 is cytochrome oxidase. What is the mode of action of dicumarol? The same as that of warfarin. Vitamin K inhibition, and that is why vitamin K was activating factor 2, 7, 9, 10, and it is inhibiting the conversion of epoxide form to its hydroxyquinone form of vitamin K, and this enzyme is epoxide reductase, and this enzyme is inhibited by warfarin and dicumarol. Which of the following reaction is exclusively performed in cytosol? Glycolysis. This requires both. This occurs in cytosol and partially in lysosomes. This requires mitochondria. A 36-year-old male presented to OPD with rashes in neck, diarrhea, and it is B3, niacin deficiency. Which of the following is not a micronutrient? Calcium. Calcium is not a micronutrient. It is required in large concentration. Its circulation is also 9 to 11 milligram per deciliter. Which of the following is most likely to cause cataract? Accumulation of गैलेक्टोज एंड ग्लूकोज. डायबिटिक्स में तो कैटरेक्ट होता ही है, एंड इन गैलेक्टोसीमिया वी हैव रिटन देयर कैरेक्टरिस्टिक बायलेटरल कैटरेक्ट, कंजेनाइटल बायलेटरल कैटरेक्ट. सो इट इज बोथ गैलेक्टोज एंड ग्लूकोज. दिस इज aldose reductase is a common enzyme which converts galactose and glucose to galactitol and sorbitol. This enzyme here is aldose reductase, and both of them are osmotically active agents. They imbibe water and they cause crystallization of lens, and hence they form cataract. Most antiatherogenic is omega 3 fatty acid, alpha linolenic acid. Melanin is coming from tyrosine. Melatonin comes from tryptophan. Melanin comes from tyrosine. Now you see a V. Now you can easily tell. Yes. If you see a cross, it will be competition. Here you see a V, it is non-competitive. In Km, same, Vmax decrease. B48 and B100 is different because of their different RNA, and this is called RNA editing. Note down, B100 and B48 are different. B100 is present on VLDL and LDL. It is different from B48, which is present on chylomicron, and this is because of a phenomena known as RNA editing. Their DNA is same, but their RNA, one change occurs, the cytosine gets changed to uracil. This phenomena is called deamination. It is deamination of cytosine to uracil, and once cytosine gets changed to uracil, this here converts CAA to UAA, and UAA is a stop codon, and hence the RNA synthesis stops. Question number 57. Nerve sensation, tingling sensation. It is B6. Here in this case it is due to vitamin B6 excess. I told you already B6 has to be maintained at a narrow level. Otherwise both deficiency and toxicity will produce tingling sensations. Which of the following is most likely cause associated with symptoms seen in excess? This is related to that question only, question number 57, and this is vitamin B6 excess. And I guess this is the last question. 52-year-old female presented to OPD with complaints of knee joint pain. She adds, as I told you, most common is not the big toe involvement, but it can be any large joint involvement. Like in this case, it is knee joint. She adds that pain is aggravated mostly whenever she sits after performing household work. There was swelling around the knee, and this is arthrocentesis of synovial fluid aspiration is done, and that reveals negative birefringent monosodium urate crystals. Monosodium urate crystals. What is your diagnosis? And this is the classical presentation of gout, except the knee involvement. The classical is actually the big toe involvement, first metatarsophalangeal joint involvement. So with this we have the quick revision on biochemistry. I have given you all the important notes. I wish you all the best. Thank you.